Microarray is a small silica or glass slide which is the size of a postage stamp that contains thousands or even hundreds of thousands of spots. Each spot contains a synthetic piece of DNA with a known sequence. A microarray consisting of partial sequences from a number of specific genes is created by placing a series of known single-stranded DNA sequences in an array on a microscope slide or a silicon chip. The sequences are placed in a specific pattern so that each position on the slide corresponds to a particular known DNA sequence. The portion of a microarray shown here only contains 6 spots, which are labeled A through F. These spots may correspond to DNA sequences found in 6 different genes. The expression of genes depends on the cell type and the environmental conditions. To study gene expression, messenger RNA or mRNA can be prepared from a particular type of cell, such as a muscle cell, or from cells that have been subjected to an environmental agent, such as a hormone. This mRNA that is expressed from such cells is used to make complementary DNA or cDNA. cDNA is made in a test tube by an enzyme called reverse transcriptase. This enzyme makes a DNA strand complementary to RNA molecules used as a template. Special nucleotides that have a fluorescent probe attached to them are used to make this cDNA. This probe will make the cDNA molecules fluoresce green when exposed to ultraviolet light. Once the fluorescent cDNA has been prepared, it is allowed to hybridize with the DNA samples spotted in the microarray. The cDNAs complementary to the type A mRNAs will hybridize with the DNA spot in the array that contains DNA complementary to the type A cDNA. Likewise, the type D cDNA will only bind to the DNA in the spot labeled D, while the type F cDNA will only bind to the spot labeled F. Since the cells did not express any mRNAs that correspond to the DNAs in the spots labeled B, C, and E, no cDNA is made that will bind to these spots. After hybridization, any unbound cDNAs are washed away, and the slide is examined under a fluorescence microscope, where the spots that contain fluorescent cDNAs can be identified. In an actual microarray analysis, a typical cell would express hundreds or thousands of different mRNAs, so that hundreds or thousands of different spots would be identified. Therefore, microarray analysis is a powerful method to study the expression of many genes simultaneously from a particular type of cell or from cells that are exposed to a specific environmental condition.